Tatiana said that she was doing it for 35 years. Does it mean that you've been doing it since the kindergarten? Because you look young. Come on. So you see the two busts, the two sculptures. Two greatest Russian scientists and scholars. Ilya Mechnikov, the recent Nobel Prize winner in biology. For more than 100 years, we hadn't had a single other Nobel Prize winner. And the second one is Professor Maximov, the founder and the first person who described a polypotent stem cell in the human body, and he laid foundation for the regenerative medicine, the use of stem cells in medicine. So it's all started off with Alexander Maximov and his scientific endeavors. Yesterday we had a slide shown to us. The last author told us that for oncologists, science is becoming more interesting than surgery, and it is truly so. Because, you know, we are talking about immunology, we are talking about cells, but basically we should have had surgeons in here, therapists, practitioners. They had to really understand what is being done and what are we doing to help them. So this slide has already been shown by previous speakers. You know, it's quite a fashionable thing to talk. It's trendy to talk about tumor editing, immunoediting of cancer, which looks like this. Dear colleagues, we can actually now launch the discussion, because I saw that there were questions to Tatiana. Can we still give the mic to Tatiana while this technical issue is being resolved?
This is my question. In your report, you said that tumor infiltration does not depend on the nosological form of cancer. Does it depend on the stage of the disease? Well, the largest specimen database for breast cancer, those are patients with primary cancer fit for surgery. There, the range is extremely versatile. So if those are primary patients, so this is the second or the first stage, so the range is from 1.2, 1.1 to 1.3. And the same stands for other exacerbated stages in third and fourth stages. For ovarian cancer, what we saw was something very specific. So now we have a specific group biopsy of normal ovarian tissue. But in the few patients with ovarian cancer, who had normally the third or the fourth stage, we usually saw a lower infiltration rate. Is it enough specimen to do flow cytometry? Yes, enough. And therefore, the marker panel is minimized. But we decided to make it as informative as possible, so as to be fit to look at small specimen for core biopsy.
So again, this is the editing schematics. You see various stages of it: elimination, equilibrium, escape, and stuff. But why the tumor still continues to grow? Or why do those tumors grow until clinical sizes where the immune system is no longer capable of combating them? 30 years ago, I was writing an article that the body needs tumors, like microorganisms, because tumors is a source of many biologically active components that are needed by the body, and without those components, the body will not continue to live, and it can control the tumor growth. Only when the tumors grow into clinical sizes, and we have millions of those tumors, in the body. We had some experiments when people died in a car crash. We would do cuts on the corpses, and we would find thousands of tumors there. It means that the immune system tracks closely every cell and kills them only when they grow onto a certain size. So before elimination, I'm pretty sure there's yet another stage, a stimulation of tumor growth. And only when immune response comes in and takes over, we see the usual situation. So imagine you have a tumor, right? And then immunocytes kill it right away. It's hard to imagine, really. There definitely must be a certain preliminary stage that stimulate the growth of this tumor. So what particular cells participate in the immune response? We have immunocompetent cells, the so-called regulatory cells, B cells, macrophages, dendrite cells, NKT cells, non-immunocompetent or formal cells, mesenchymal stem cells, suppressors of myeloid nature, two types of them, MO and GR, stromal fibroblasts. They produce homosuppressive action, and endothelial cells, adipocytes, granulocytes. And it was also fixed back in time. It is our recent discovery of our institute. Erythroblasts have a very powerful immunosuppressive symptom. T suppressors. It's been calculated how many subpopulations are there. There are more than 10 of them. And all of them, more or less, produce this immunosuppressive action. CD4, CD25, plus, that's what is most interesting for us, DN tract, tier 1, tier 3, CD8, CD28 minus, sometimes we mention them. Others are not that noticeable because they're less active in increasing the tumor. So TGF beta, IL-10, PGE2, and IDO. This is what produced by those suppressors. B suppressors, we have various phenotypes. There's a phenotype type B that has immunosuppressive actions, and it induces interleukin 10, interleucin 10. B cells participate in cancerogenesis. It means that lysine is a very powerful immunosuppressive factor that comes in force during cancerogenesis, and Tregs are also produced there. Macrophages. It's already been told that we have mainly two macrophages, and we are currently observing the third population. But M1, M2 macrophages are clearly related to immunosuppressive activity. When tumor grows, they are accumulated in the tumor itself. They suppress a whole number of factors. PG, IL-10, and so on and so forth, TGF beta, everything. So these are the responses they produce. And I would like to mention here that there's four major immunosuppressive factors here that produce macrophages, that suppress macrophages. Dendrite cells. Dendrite cell ensures stable existence of Treg inside the tumor, and they produce their suppressive activity. Dendrite cells, macrophages produce IL-10. They ensure the accumulation of cells with immunosuppressive properties. These cells, dendrite cells, may induce a Treg accumulation that produce IL in return. So they lead to a very powerful immunosuppressive action. And you can see it's schematically how it's all done. And those dendrite cells, they produce there's a very powerful molecule that we believe is in the core of all immunosuppressive mechanisms. Mesenchymal stem cells. For the first time, they were described by Isaac Furnistein, a Russian scientist, a Russian scholar, and his works laid the ground for this research of regenerative medicine, and now they're being used at various pathologies when treating them. And it turned out those mesenchymal stem cells have an immunosuppressive effect. We know that normally they live in bone marrow and they don't have this immunosuppressive action, but as soon as inflammation starts, gamma interferon is produced, and that in turn inhibits proliferation and induces apoptosis. And today, mesenchymal stem cells are very often used in clinics, not just by ourselves, but everywhere in the world. And here are the molecules TGF-beta, IL-10, PG2, IDO, and a number of others that synthesize mesenchymal stem cells when they become immunosuppressive. Here I'm showing the actions. So you see it's active across various directions. Macrophages, dendrite cells, mesenchymal cells, they act on many immune mechanisms. By suppressing them, they ensure the tumor growth. We're saying that they induce immunosuppressive activity at macrophages. Dendrite cells stimulate this, and you can read it for yourselves. So these are 4 factors, and I'm going to refer to them all the time because these are very important now. Myeloid suppressing cells. Basically, these are those cells that appear at the differentiating stage. Granulocytic macrophage array. These are the precursors of macrophages, and we have basically 2 types of suppressor cells, granulocytes and lympholytes. Same mechanisms. They produce arginase that cleaves arginine. Basically all the same immune suppressive molecules. Again, I'm showing it schematically, and then the macrophage acts on myeloid-DSC. They stimulate macrophage in response. Macrophage then produces PGE, and everything goes in this circle. Erythroid suppressor cells. For the first time, we managed to identify those cells. We showed a humoral mechanism of the suppression function, and we identified the possible application for them. They have cytotoxic feature, and they are related to a whole range of various tumors. And NKT cells, lots has been said about them. They are definitely they defined as a subset of T cells that expressed the natural killer, and it's now generally accepted that they participate in this immune response as well. They have this stromal cells, endothelial cells, stromal fibroblasts of liver. There's so many cells that are non-immune competent, but that have a capacity of suppressing immune system cells. What kind of mechanisms are there? Well, they develop regulatory T cells, they ensure accumulation of suppressor cells, macrophages participation, and molecular mechanisms: IL-10, TGF-beta, PG2, IDO, and arginase 1. I will add just to that list. The most dangerous, the most scary thing, here is that all of those cells are actively interacting with each other. We are trying to take out Treg, that's what we're doing, and we see certain effect of it, but they will never be able to get to the end of it, because the suppression activity is continued by, say, myeloid cells, macrophages. They continue doing the suppression, and they're very active in suppressing it. Myeloid cells will continue to stimulate Tregs growth. So there's always a constant interaction between those basic elements, those basic bricks in this wall. And of course, myeloid suppressor cells, they induce a macrophage accumulation of M2 phenotype in the tumor. B1 regulatory cells induce macrophage accumulation of M2 phenotype by means of producing IL-10 and so on and so forth. Once again, I would like to reiterate that there is a constant interaction, there is a constant communication, which is positive for the cells, but which is negative for our body, right? All those suppressive populations, they continue to talk with each other. Macrophages induce myeloid suppressor cells and various mechanisms, and you can read it all again, all over again. We're saying that basically, just by targeting, just by trying to get rid of one suppressive population, we will never be able to get rid of the whole process. Moreover, the tumors, they synthesize a whole range of various elements, immunosuppressive, inducing, suppressing cells, interleukin 10, Cytotoxicity 2, and they induce T suppressors and they suppress effector cells of the immune system. So we're showing what kind of cells are synthesized. Interleukin 10. Many cells produce it. IDO-PG2 is synthesized by macrophages, dendrite cells, fibroblasts, and so on and so forth. So what is IDO? Indolamine 2,3 dioxygenase. A lot of attention is being paid to it. It is said that this particular IDO plays a very important role in suppressing. This is it cleavages tryptophan. It sits on the membrane, and in the surrounding media, it cleaves leptophan to kynurenine and further on. The change in the level of tryptophan is already a very powerful immunosuppressive factor. And you see the metabolism here of tryptophan, and there's a whole range of metabolites that have a very powerful immunodepressive effect. Gamma interferon. Any increase of gamma interferon will lead to expression of this catalyst, of this enzyme, and it will lead to tryptophan hunger, and those metabolites of tryptophan will appear. They have immunosuppressive effects, so it's all connected. Well, there are certain IDO inhibitors, not yet in the clinic, but one of the most powerful inhibitors here is 1-methyltryptophan, but today we're just experimenting with that. Cyclooxygenase and PG. I found an article in a magazine which was distributed in here. Americans say that we need to use aspirin, not just for preventing colorectal cancer, but it's been said that if people would take aspirin on a regular basis, they would have a rectum cancer much rarely. And then they describe the effect, the action. Quite often, this PG aspirin turns cancerogenic and aggravates the tumor. Americans and British said that basically aspirin has to be used not for preventing cancer, but to treat cancer that's already there. And it has to be noted that aspirin, PGE, is a very powerful immunosuppressive factor. There are two things here. Cyclooxygenase and PGE, they are responsible for immunosuppression because all effector cells subjected to the suppressive influence of these two things, tryptophan, hunger, and PGE2. They are very, very powerful things. They're very powerful suppressive factors. And what I'm saying in here is that PGE acts on everything. It reduces the macrophage production in dendrite cells, cytotoxic T lymphocytes. Everything is influenced by prostaglandin. Tumor cells themselves can synthesize PGE2, and that serves as a yet additional factor of growth. And PGE, for a number of tumors, is also a growth factor. It means that it can sustain itself. What immunotherapeutic methods are available right now? And we're showing it here, vaccines. We'll be talking more about it. And oncolytic viruses, adaptive cell therapy, immunocompetent cells, effectors, antibodies against tumor antigens, against growth factors, and the so-called checkpoint blockade, a very trendy expression and a buzzword, and combined immunotherapy. So, what kind of molecules can serve as a checkpoint? You know, lots is said about checkpoints, and probably it does deserve our attention. Same IDO. I told you that this enzyme sits on the membrane of a macrophage. It suppresses. It is a myeloid nature suppressor, and our body produces CD8 plus cytotoxic T lymphocytes. Once a Nobel Prize was given for this, actually, for this theory. Probably the same mechanism takes place in here. When there's not enough of something in the body, the immune system would not react to it. But as soon as this enzyme, well, it seems like it's natural, right? It's what you produce naturally, but as soon as you get more of it, the immune system starts reacting, starts responding to it. And then the immune system finds certain lymphocytes that have cytotoxic capability, cytotoxic 2 cells that express in such a way. Tumor cells express it this way, and dendrite cells express it this way. They express this IDO, and it has immunosuppressive effect. And our body starts working against them, and we are planning to produce a population of cytotoxic population of these cells in our institute in order to see how this whole effect is rendered. And we're showing it schematically, how the cells are being produced, how they germinate in vitro, how this lymphocyte is produced, and then it's injected in the body with the single purpose to kill those cells. And we are showing that if you use this vaccine, you can see a reduction in Tregs and more cytokines. Same. As for cytooxygenase, we haven't found any cytokines, but we produced antibodies that could work against this enzyme. And as I told you already, we saw that they can have this very powerful effect. Maybe we will find in the end those cytotoxic lymphocytes that influence it. FOXO3 transcription factors, FOXP3 transcription factor, they localize suppressor cells, same. We found in the body cytotoxic lymphocytes that can work against them. Again, our body, when something grows quite intensively, when suppressing cells are becoming very active, well, then you will get this. FOXP3 is metabolized in the cell, and it goes outside the cell and starts producing cytotoxic lymphocytes against T suppressors specifically. And I think we'll be able to produce lymphocytes that have this cytotoxic capability against Tregs. We can do this way. And we're showing you how it can be obtained and what kind of effect we can achieve by applying it. Next target, next checkpoint. It's an antigen that sits on Tregs. It sits on many cells, but Tregs are the ones which accumulate them. And against this. Well, there are antibodies that can work against it, and right now there are no cytotoxic lymphocytes that can work against it. We don't know why. It hasn't been described, but there's another factor that also characterizes suppressive activities of Tregs. It's called PD-1, or its ligand, PD-L1, and we found lymphocytes. So there are lymphocytes against it, of course. There's a regulatory mechanism in the body. When you have too many, too big a level of those factors, cytotoxic lymphocytes will start producing. It's the same approach. That's the way you can get those lymphocytes. In vitro, you can produce them in needed quantities, then you just re-inject it back, and in this way you suppress the Tregs activity. Of course, if you just use one antibody versus a particular population or versus anti-CTL4, you will obtain a certain effect, but it won't be optimal. You need a combined, you need a package of various antibodies. So when those antibodies were used against CTL and PD-1, together the effect was much higher. The effect was much bigger. It was stimulating the immune system. It was much better if you just used one antibody and that's it. There are some elements that also influence the expression. You know, resveratrol is a very popular now thing. It's a very popular molecule that's been produced from red wine. Academician Chazov was always saying that the best anti-atherosclerotic drug is a glass of red wine every day, and this resveratrol is a molecule produced from red wine. It's been described quite extensively in the literature, and it does have antisuppressing effect, and it can work like an antisuppressant in many stages. And there's another very popular and a very fashionable molecule. It's a herb, curcumin, and all the substances produced from there. It's already said in the literature that it can influence the suppression of cells, and it can suppress the tumor and the tumor cells, and it can stimulate in parallel your immune response. Some of the drugs based on curcumin are already available, are sold off the shelves in Europe and in Russia as well, and you can still buy it in drugstores. Aspirin, back to basics. As I told you already, that aspirin is probably the most powerful drug. It's been out there for more than 1 century, and I don't know how many lives it saved. You know, it has anti-inflammatory properties, but of course you have to combine it with all those anti-tumor drugs because it suppresses PGE molecule that has a very powerful monosuppressive activity. This schematic shows that it can inhibit IDO as well. We need to use the engineering approach, an engineering approach to oncology. And we need to develop a whole network of immunomodulating cocktails that will ensure better treatment. Thank you.